Thanks, Perry, and good morning, everyone. Hopefully you can all hear me. I've been asked to talk about rust incursions, and to give you some idea of why I've been asked to speak about that, well, in the context of myrtle rust at least, my journey with plant pathology started 45 years ago, not working on in agriculture, but in fact working in forestry. I did my PhD honours and PhD on some foliar pathogens of eucalypts known as Mycosphaerella. I think they're called Teratosphaeria now. They're always undergoing taxonomic changes. But anyway, I spent 6 years of my life doing honours and PhD on these pathogens. I then tried to secure funding to go to Brazil on a postdoc to work on myrtle rust. So this is back in the early 80s, mid-80s, and sadly that funding application was unsuccessful. So I was then trying to secure a position in forestry. There were none about, so I ended up changing and working on these things, cereal rusts, and I've been doing that ever since. So that's now 38 years, I think, that I've been working on cereal rusts. So my talk today is going to focus mainly on cereal rust. We've got very good understanding of variation in these pathogens in Australia in the context of exotic incursions. So they provide very good examples that can be used and applied to other systems like myrtle rust. Okay, so Australasia, an isolated region from other parts of the world, as we know. If you've come from overseas, I always say to people, if you've come from the Northern Hemisphere to a meeting here, then you know this because you've had to sit on an aeroplane for an awfully long time to get here. So very, very isolated. In the context of cereals, we know from our long-term surveillance that not only Australia but New Zealand forms pretty much a single epidemiological unit. We've got many, many examples of the inoculum exchange of cereal rust between Australia and New Zealand over the years, and sometimes that's been as quickly as 1 year. So we've detected a new strain of rust in Australia, wheat rust, and within a year it's been present in New Zealand. So in some ways, I was a little surprised that it took so long for myrtle rust to appear in New Zealand, given that previous experience with cereal rusts. We know we've had periodic incursions of exotic rust isolates, cereal rust isolates, over those many, many years we've been monitoring them. But, you know, and of course they've had huge impact, and we see that now also with myrtle rust. So this is very unfortunate, but on the positive side, if I may try and put a positive spin on it, when we do get these incursions, it provides a very rare opportunity to undertake studies of how these things spread and how they evolve because we are an isolated region. So we can look at continental scale evolution in these rust pathogens when they come in. And many of them, in the case of the cereal rust and also myrtle rust, we believe, are clonal, they're asexual. So again, we can look particularly at the processes generating genetic variation in these pathogens in the absence of sex. And I've said there that, you know, we've got long-term studies for some of these cereal rust pathogens of over 100 years. So quite remarkable, really, that here at the University of Sydney back in 1921, Professor Waterhouse began monitoring races, strains of wheat stem rust and wheat leaf rust of wheat. And we've done that every year since. So we're now well over 100 years of continuous monitoring of these rust pathogens in Australia. And we preserve isolates of these rusts in liquid nitrogen. We've got a very extensive collection of them, a historical collection, extremely important nowadays for rust genomics, but also allowing us to do the research that we need to do to help us confirm exotic incursions. You know, when you get an isolate come in, how do you know it's come from overseas? You need to do the basic pathology and genetics to show that it's actually different to what we've got. So those reference isolates are critical that we've kept. Detecting those new pathotypes or strains, if you want to call them, the ones that arise locally, and determining how they arise and what impact they have if and when they do arise. Assessing their impact and warning the industry. So this is a very important part of what we do. Yes, it's new, but what does it do? That's what the industry wants to know. What is its impact, and what direction should our resistance breeding now take as a result of this new incursion or new pathotype? And as I've already made the point, preserving these rust isolates is just absolutely critical to have that resource. It's just like having a collection of any biological thing, seed collections, whatever. These pathogen isolate collections are absolutely critical for the research that we're doing to help our industry produce cereals more sustainably. So what have we learnt from that from those long-term rust surveys? So this is wheat rust going back to 1921, and it shows 15 incursions that we've detected over that period of time. So over 100 years, we've picked up 15 exotic incursions of wheat rust, and the most striking thing in that graph is that it's going up, right? And it's not because we've got better surveillance methods these days. You can see back in these are 20-year incremental periods that I've broken it up into. You can see back in the 1955 to 1974 20-year period, nearly 19,000 samples were analysed of wheat rust, were analysed for strain analysis, and only 2 exotic incursions were found. You come up to our current 20-year period, started in 2015, we're 8 years into that. We've only looked at 2,500 isolates. We've already found 3 incursions, 3 exotic isolates. So this is real. We are really seeing, we've seen an uptick in the frequency of the incursion of these exotic rust isolates. And why is that? Well, we have examples from the late 1960s of rust movement from southern Africa, southern and central Africa, into Australia with high altitude winds. We believe that's how they got here. And of course, we've got many examples of rust moving from eastern Australia into New Zealand. But we believe that this increase in frequency in incursions is due to people walking these in inadvertently on contaminated clothing. So that's my boot. From a few years ago when I was taking notes in one of our rust nurseries, we assess a lot of breeders' material. I think this year we'll have probably 150,000 breeding plots that we're assessing in the field. We go through, we artificially inoculate them with defined pathotypes that we get out of a collection, so that the information we generate for the breeders is relevant to what's going on out there in the agricultural areas. And we go through and assess those plots, and we get covered in rust. And those rust spores remain viable, of course, for at least 2 weeks under viable conditions. If you don't wash your clothes, you're going to walk them around somewhere. So I was involved for many years, I think it was 8 years, in a very important project funded by the Gates Foundation in East Africa on a stem rust isolate that emerged there, UG99, and I was absolutely paranoid I was going to bring that thing back with me. So I used to leave clothing in East Africa, my boots and stuff, and wash things about 5 times because I know about this, and I was absolutely paranoid I was going to bring that thing back to Australia. All right, so here are the 15 documented cases of exotic wheat rust incursions that we've had in Australia going back to 1925. The first one was found then by Professor Waterhouse. And we've got isolates of these, representative isolates of all of these, viable isolates of all of these in our rust collection. And we've been back and we've been sequencing them progressively and getting a better understanding of genetic variation and also where they came from. So you'll see in that last column there, possible origin, lots of question marks. We're now starting to fill those question marks in. It was difficult to say in the past just based on virulence. The sequence data is now giving us a much better understanding of where these things have come from. And of course, that can give us an understanding of potential entry pathways. Those 2 in 1969, Central Africa, and the one in 1954, we believe all came to Australia on high altitude winds. The ones I will talk specifically about are these four incursions of wheat stripe rust. So there are 3 rust pathogens that infect wheat. Leaf rust and stem rust we have had present in Australia since at least European colonisation and possibly before. There is a native grass that's infected by both of those. But this guy, stripe rust, the third one, not detected until 1979. And 2 years later, it was in New Zealand. And it's caused a lot of problems over there. And still does. So it first turned up in 1979. We believe that incursion came from Europe. We then had a second incursion into Western Australia, and within a year that was present in Eastern Australia. That came from North America. And then 2 very recent incursions, 2017, 2018, and these have caused real headaches for our wheat growers. Last year in Eastern Australia, we probably had our worst wheat stripe rust epidemic on record, and it was caused by those two incursions in 2017, 2018. I did the race analysis on that. We looked at 800 samples from last year. 99% of them were, I shouldn't say 99% of them, but the vast majority of them were one of these two incursions from 2017, 2018. How do we know where they've come from? Well, we're doing whole genome sequencing. This is just a phylogram showing 10 representative Australian isolates of that wheat stripe rust pathogen, including those 4 incursions, and 315 non-Australian isolates, and we've been able to fit them into a global phylogeny, and that's been allowed us to trace them back to figure out where they came from. I'm being told I've got 10 minutes, 5 minutes, 5 minutes? Okay, so I'll quickly go through the next three just to show you what happens when these things turn up. This is the 198 that came in 2018, drought year, picked up in southern New South Wales. By the end of the year, in a drought year, that had been detected in Tasmania and as far north as around the same latitude as Sydney. The following year, a drought year, it extended its range even further. And then in 2020, when we returned to normal seasonal conditions, it was everywhere. And again, last year, it really caused some headaches for our growers in Eastern Australia. So these things move around really, really quickly once they get in. We can look at long-term population structure. So this is stripe rust. We've basically got 5 genetic groups of wheat stripe rust in Australia going back to 1979. You'll see all the blue colour to start with. We had the incursion in 2002. The blue clade almost disappeared entirely. The red one overran it. And then in more recent years, we've got this more diverse population. And in fact, last year, real genetic diversity in our pathogen population of stripe rust. This makes the resistance breeding far more challenging and also providing extension advice to farmers as to whether they should spray varieties or not. Those 4 incursions have caused massive economic damage to our agricultural industries. The one in 2002, up to $90 million spent in fungicides. The two most recent ones, I'd say last year, I'd say $100 million spent on fungicides in Eastern Australia would not be unrealistic. All right, so they've had huge economic impact. The other thing is we always look at virulence, and yes, this thing can attack a previously resistant variety or a plant, but what we're also finding is these incursions are bringing in other traits. So we've detected probably the first 2 confirmed cases of fungicide insensitivity in a rust, and both of them are incursions. So these incursions are not just bringing in new virulence, they're bringing in traits like fungicide insensitivity as well. To illustrate that, I'm running out of time here, but this is 3 isolates. We've tested these leaves. We've applied different rates of fungicide to these leaves, increasing from left to right. 3 old isolates, all sensitive. More recent one, insensitive. We've done a trace back on that, traced it back to an exotic incursion. This is barley leaf rust in 2001 using our collection. So that was found in Western Australia, that incursion, fungicide insensitive, spread to the east, and it's everywhere. So it's also undergone sexual recombination. It's the only rust we've got in Australia that undergoes sexual recombination. It's recombined that fungicide insensitivity with virulence. So just to conclude, I hope I have a minute to some concluding points. Talk about 2 minutes. Great, I've been going way too fast, eh? So 100 years, more than 100 years of monitoring cereal rust pathogens. This has underpinned all of our efforts to control cereal rust in Australia, right? It really is fundamental. If you don't know what you're dealing with in terms of the pathogen, you cannot breed efficiently for resistance. You cannot provide advice to growers on whether they should use fungicides or not. You must know the virulence and the impact of those rust isolates on current germplasm. This has enabled us to make tremendous progress with controlling these pathogens in agriculture. So it's been estimated that just in wheat and barley alone, that our work, that's all underpinned by these race surveys and monitoring these incursions and working out their impact and allowing breeders then to use that information to redirect their resistance breeding efforts, over $1 billion saved a year. That was an analysis done a few years ago by some economics people. But where we've been less successful, so this has been in dealing with the locally emerging rust pathogen isolates that have emerged through mutation and processes like that. But where we've been far less successful is dealing with these exotics, because we don't know when they're going to come in, we don't know where they're going to come from, and we don't know what attributes they're going to bring with us. So they've had massive impact, these exotics, on varieties and on breeding germplasm. So don't forget that. You get an incursion come in, yes, your current varieties are affected, but then you've got these breeding programs that invested a lot of money. It takes about 10 years to breed a new wheat variety, and they've got to throw a lot of that germplasm out. So it has a huge impact also on breeding programs and slowing genetic gain. But we're also seeing other traits come in, like insensitivity to fungicides, as I mentioned. So it's not all about the virulence. Sometimes these things bring in other things. And one thing I didn't mention is that we've had isolates of striped rust come in that are higher temperature adapted and more aggressive. And this has changed the footprint of this disease. So it's not all about the virulence. What I will say, and I think this is relevant to Bob's point this morning about the importance of national coordination. For our program on cereal rust, this has been absolutely critical, right? We just have not been able to do what we've been able to do without national coordination and long-term commitment through our grains industry and the University of Sydney. And I'll make one final point, and that is that these diseases are not going to go away. And then without that commitment, we can expect more impact in future. We need talented people, we need to retain the skill base, we need funding, and if we don't get that with the coordination, we're going to have problems in future. Thank you.